So yeah, this is welcome. This is our first seminar of the fall 2024 SNR seminar series. Thanks for coming. Before we get started, the seminar class, we have 2 students, and I assume it's you guys, right? Okay. And yeah, I think that's all we have. So next week we'll have another seminar at 3:30 on Wednesday. It's going to be Laura Fowler from Penn State. She'll be talking about sustainability and specifically the role of universities in helping to find solutions for sustainability. We good there? Okay, well, today we have a speaker that's relatively new to the SNR community, but I'll let John Spurgeon introduce the speaker.
So today I'm pleased to introduce Clint Leach. He has been here for 2 weeks, about 2.5, 3 weeks, not too long. He's a new assistant unit leader with the Nebraska Cooperative Fish and Wildlife Research Unit. He is our new statistician, stats assassin, and we're really excited for his skill set here at the unit and within SNR. I know I'm looking forward to him building relationships with all of our cooperators and doing some great things. Clint got his master's degree in statistics at Colorado State and his PhD in ecology at Colorado State, and also followed by a postdoc. Bayesian is the word, I do believe, when it comes to his research, and pushing the boundaries of how we can better manage and utilize our data that we collect and from all these messy studies. So with that, Clint, take it away.
All right, thanks John for the introduction. Thanks to John Benson for inviting me to give the seminar, and for you all for being here. I appreciate the opportunity to introduce myself to the department here and talk a little bit about my work. This will be very similar to the interview talk I gave back in the spring, if you were present for that. So we'll cover some of the same territory, but maybe with a little less anxiety this time around. I'm going to try to manage 2 devices at once here, the microphone and the pointer. We'll see how it goes. And of course, the pointer worked before. No, it doesn't. Okay, I will not manage 2 devices. Oh, I will manage no devices at all. Okay, so start out with just some acknowledgements. All the work I'll be talking about today was done with a number of collaborators, and it was funded by the Southeast Alaska Inventory and Monitoring Network, as well as the Glacier Bay Marine Management Fund. It was done in close collaboration with Mevin Hooten, my postdoc advisor, as well as Perry Williams and Joe Eisiger, and a large group of scientists up in Alaska at the USGS, U.S. Fish and Wildlife Service, and National Park Service. So all of the work I'll present today owes a lot to those folks, and they're the ones who actually were out in the field collecting all of the data that I'll be discussing today, because that's not my job. So my research in general focuses on monitoring and managing population spread. That's both in the context of monitoring and managing recovery, so species that have been formerly extirpated from their historical range, monitoring their spread and recovery back into that range. So in the context I'll discuss today, that's looking at sea otters in the North Pacific and their recent expansion there. And then on the flip side of that is monitoring and managing invasion, where we have an introduced species that is now expanding into novel habitats. So pulling an example from my work, looking at brown tree snakes and tree snake movement and spread in Guam. I won't have a chance to talk much about that today, but I'm happy to chat about it later if anybody's interested. So in both of these cases, we're interested in sort of capturing these complex spatiotemporal processes and capturing how these species are interacting with and changing the communities that they're moving into. And so this comes with a lot of quantitative challenges that go along with it. One of those being that these data are often very costly to collect. So this picture here is an image taken from a sea otter aerial survey flown in Glacier Bay. So these aerial surveys, flights are very costly, time consuming, and potentially even dangerous. And so there's a real incentive to really make sure that we are optimizing the amount of information we can get out of these data and make sure we're using them appropriately. And to take an example from the ground, animal telemetry data are also often very challenging and costly to collect. This is my colleague, Abby Fuca, out traipsing through the forests on Guam trying to track down snakes. And so these data also come with a pretty large price tag, and it's important to try to use them effectively and efficiently. In addition to that, we also are often dealing with multiple data sets. Often the inference that we want to get for a particular question requires leveraging multiple data sets. This idea has been really well developed in the integrated population modeling literature that was sort of kicked off by Schaub and Abadi. And in these contexts, we're often dealing with multiple data sets on a given population. We might have population count data along with demographic data from, say, a mark recapture type study, and we need to bring those together to get inference on a shared process. But in other settings, we might, like in multitrophic settings, we may have to deal with data sets on different species, and potentially when those data sets weren't necessarily collected as part of a single unified study, but we still need to try to bring them together to understand the ecological processes. And then lastly, especially in management settings, we really want to make sure that we're getting interpretable inference about ecologically meaningful quantities. So this is an example here, again with sea otters in Southeast Alaska. And so this is work done by my colleague Joe Eisiger, modeling or estimating the actual spread rate in kilometers per year of sea otters across Southeast Alaska. So getting these kind of real tangible quantities out of our models is often important in these sort of management settings. And so as John already mentioned, my approach for dealing with these things is Bayesian hierarchical modeling. This is a super useful framework in large part because it lets us partition a given problem into these different levels. So we can break it up into the data level, the observation process, and anything we need to account for there. Then the process level, where we actually capture the sort of latent, unobserved ecological process. And then lastly, all of the parameters that go into all of that. And this is particularly useful for linking multiple disparate data sets together and for modeling how those different data sets might arise from a shared ecological process and capturing how that process can give rise to these different data streams. The Bayesian approach is also really useful for leveraging existing ecological knowledge and bringing that to bear on these questions. In particular through the use of priors in Bayesian analysis. That provides a natural place to incorporate existing ecological knowledge that we can draw some strength from, but then also in the process level of the model where we actually specify our hypotheses for how we expect the system is, the rules that we expect to be governing the system. And in particular, I think differential equations provide a really valuable tool and a really powerful tool to use here. Differential equations provide sort of a natural language to describe how we expect a system to evolve in space and time. And so embedded in this sort of larger framework, the expressiveness of differential equations can be really powerful. And then lastly, I'm also interested in taking all of the learning and information that is embedded in these Bayesian hierarchical frameworks. This is often a decent amount of work to put this together and fit it to data. And it's, we can get really useful inference out of it, but I'm also interested in taking it the next step and using it to inform the collection of future data, sort of closing the loop on the scientific process here of using this to optimize our future data to kind of feed back in and learn efficiently in the future. So that's kind of a general overview of kind of my approach and motivating questions. So getting into some specifics of how I've implemented this, I'll start off with this multiple data set context and linking those multiple data sets together through these mechanistic models. And I'll talk about this in the context of sea otters, again, in the North Pacific. So shown in yellow here is the historical range of sea otters. So they historically have covered, most of the coast of the North Pacific. Then due to the maritime fur trade, they were extirpated to all but just a handful of remnant populations shown with these red stars. But more recently, in the last several decades, due to legislative protections and reintroductions, they've been making a pretty impressive recovery across much of this range. And so this purple here is showing their current range that they've recovered from these handful of remnant populations. And so we'll be talking specifically about Glacier Bay here in a few slides, and just to orient you, that's about right there. So the reason this recovery is important to understand and to kind of monitor and manage is that sea otters are a well-known keystone species. They're voracious predators. They have to eat about 25% of their body weight daily to maintain their metabolism, so they can really exert a strong top-down influence on the communities they occupy. And this has been really well developed in the rocky intertidal systems, where there's this sort of classic trophic cascade that we observe, where in systems with otters, the otters suppress sea urchin herbivores, and that enables kelp to thrive and kelp forest ecosystems to form. But in systems where otters are absent, those sea urchins erupt, they graze down all of the kelp, and we kind of tip the ecosystem over into this alternative state known as urchin barrens, where we just kind of have a carpet of urchins and not much else. So we expect that as sea otters are recovering across this former range, they're going to bring a lot of changes with them. So that brings us here to Glacier Bay. Just to orient us, here's the Gulf of Alaska, and this big Y-shaped water body here is Glacier Bay. And so Glacier Bay, as late as the mid-1700s, like 1750 or so, was entirely covered in ice, and then it has rapidly deglaciated since. And so the nearshore benthic community of Glacier Bay has developed pretty much entirely in the absence of sea otters. And so sea otters then arrived here. They arrived at the mouth of Glacier Bay from reintroduction efforts in the early 1990s, and then have since spread across most of the bay and have reached a total population of somewhere around like 8,000 or potentially more. So they've been very successful here. And Glacier Bay is a mixed sediment habitat predominantly. So rather than sort of the rocky substrate that the kelp system grows on, this is more of a sandy substrate where clams are one of the main prey group for sea otters. And so we're interested then in understanding, as sea otters spread through Glacier Bay, how are they affecting this community that has existed in the absence of this kind of apex predation. Specifically, we're going to focus on butter clams. So this is like a large clam that's one of the favored prey sources for sea otters, and want to look at how sea otters are changing the distribution and abundance of butter clams in Glacier Bay. And to do that, we have two sources of data we can leverage. We have sea otter aerial surveys, and we have intertidal and subtidal clam surveys. So we have both pieces. We have the data on the otters, we have the data on the clams, and we need to put those two together. The challenge here is that these data sets are not necessarily aligned in space and time. So this plot here is showing these are all of our prey sampling sites along the x-axis. These are years along the y-axis, and so each of these points represents that year was sampled, or that site was sampled in that year. And then the horizontal lines here indicate years that sea otter aerial surveys were flown. The dashed lines correspond to sort of a broad distributional survey where they just fly the coastline and try to identify the edges of the sea otter distribution. And then the solid colored lines are when design-based transect surveys were flown. And so we can see there's a lot of overlap here, but we also have some mismatch. We have sites that were prey sites sampled in years without sea otter surveys flown. So we don't always have just like a paired data of sea otter abundance to go with our clam abundances. And we'll also note here that the first prey sampling wasn't done until 1998, 1999. That's still relatively early in the sea otter expansion into Glacier Bay, but it's still not quite early enough to represent like a true pre-sea otter state. So we need to account for that as well. When we have a similar challenge, when we look spatially too, this is a snapshot of a given year, 2006. These X's show the locations of the prey sites that were sampled in those years. The horizontal gray lines are the sea otter aerial transects that were flown that year. So sort of like we saw on the temporal side of things, we have some overlap where we have both prey data and sea otter data, but we also have a lot of prey sites that take place that were sampled outside of the domain of those sea otter surveys. And so if we want to try to line these things up and get sea otter estimates that go alongside our clam data, we need to do some sort of modeling here. We could potentially just do some sort of interpolation or Kriging type approach just to kind of smooth out the sea otter data and get estimates at our prey sites. That leaves a lot of ecological mechanism out. And those estimates maybe don't agree with sort of the physical principles we might expect guide the diffusion of an animal across this landscape. So what my colleague Lucy Liu did in 2018, she developed this sea otter ecological diffusion model. And so I'll just walk through this quickly. So in this approach, we take those sea otter transects, and those have a observed width of about 400 meters. And we chunk that up into 400-meter grid cells. And then we can model the count of sea otters in grid cell I in year T as arising from a binomial distribution with a true latent number of total sea otters in that grid cell N and a detection probability P. And then that latent true abundance arises from a negative binomial, and the negative binomial is parameterized to have a mean governed by this spatial temporal density process lambda. So lambda ST is the density of sea otters at location S in year T. And that then is governed by this partial differential equation. So this is an ecological diffusion model. So the rate of change of the density of otters at location S in year T is, there's this diffusion component that kind of is what it sounds like it is. It's just the diffusion of sea otters in space, where that diffusion rate can vary spatially based on different environmental features. And then locally, at a particular location, we expect local logistic growth. And so the main thing to take away here is we can fit this model, and then what it gives us is it gives us these spatio-temporally complete maps of sea otter abundance, or of estimated sea otter abundance. So this is showing one of those maps. Limited the map here just to show the near shore, so everything with a depth of 40 meters or less. And so at each of these sites, our prey sites indicated by an X, now we have this complete time series of estimated sea otter abundance that we can use. And so from there, then we can actually get to modeling the clam dynamics. And so the ZJIT here is the count of clams observed in quadrat J at site I in year T. So these are 1/4 meter square quadrats that they're placing along a transect at each of these sites, excavating to a depth of 25 centimeters, and then counting and identifying all the clams that they find there. So that's what the raw data are here, and we again model those as a negative binomial with a mean determined by this latent true abundance, NIT. And then again, we're gonna play the differential equation game and specify the evolution of this NIT with a differential equation that governs recruitment. And we'll say that we have spatially variable recruitment a background mortality, and then lastly, sea otter predation. So here, this lambda I hat, that is the posterior mean abundance of sea otters at site I that we derive from that ecological diffusion model. And then we also parameterize it with this A, which is a spatially variable attack rate parameter to capture the strength of sea otter predation at a given site. And so we fit that model and we can estimate dynamics at each of our prey sites that look something like this. So this is clam abundance per quadrat through time. The black points are our observed quadrat counts. And then the black line and ribbon is our posterior mean and estimated credible interval. And we can get a plot like this at each of those red X's, each of our prey sampling sites. And so we can, this captures, how the, as sea otter predate, the effects of sea otter predation accumulate through time, the abundance of clams declines. And a key point here is, again, we don't observe any of these clam data prior to 1999. And because the differential equation offers this really natural way to integrate the effects of sea otter predation forward through time, we can actually sort of walk it back and estimate the initial clam abundance in 1993 before sea otters had spread into the bay, and then capture how that had changed by our first data observations and how that carries forward through time. And what this lets us do is estimate the environmental drivers of clam abundance prior to the effects of sea otter predation. So that's what these figures are showing. We have log clam abundance per quadrat as a function of a few different environmental drivers. And so prior to sea otter arrival, we see a slight positive latitudinal gradient in Glacier Bay. So as you move up the bay, clams are becoming more abundant. And we see this kind of unimodal relationship with current speed. So there appears to be this sort of sweet spot of current speed where that's most suitable for clam habitat. And then we can capture how that changes after 25 years of sea otter predation. So these blue lines now are showing the estimated environmental gradients in 2018. So sort of the post-otter or mid-otter state. And so we can see that otters really are restructuring the clam abundance and the spatial distribution of clams in Glacier Bay. Sort of as they're eating their way up the bay, they're making the latitudinal gradient steeper. And they've also eroded this peak in clam abundance as a function of current speed, potentially because they're finding these really good clam habitats and preferentially grazing them down. So we don't really see that same shape. And so this framework then has let us capture and start to understand how clam abundance is changing in Glacier Bay. And on the other side of it, we can also look at attack rate, sea otter attack rate, or predation intensity as a function of environmental covariates as well. And we see that as a function of length of shoreline. So this is kind of a measure of shoreline complexity in the vicinity of a given sampling site. So as the length of the shoreline in the neighborhood of a site increases, that's sort of a windier, more complex shoreline, or potentially like a protected cove or inlet. And so we can see that those sites with the most complex shorelines have a lower sea otter attack rate. So potentially sea otters are not getting into these more sheltered areas, and those potentially could serve as sort of a refuge for clams going forward. Okay, so that covers the sort of multi-data set context. So we'll pivot now to the future data set and thinking about how to use these hierarchical models to inform the collection of future data. So the motivation here again, we'll go back to this plot from Glacier Bay, is, this is a long-term survey that they're conducting in Glacier Bay. They're going to keep flying these transects every year, every several years as feasible, and they need to decide how to allocate this effort. We can only fly so many transects in a given summer, so we need a way to choose what transects we do fly intelligently. And so in order to do that, we have to first set our goal and determine what we're trying to accomplish with the study, the monitoring program. And so in Glacier Bay, what we really want to do is try to understand the total abundance of sea otters in Glacier Bay. That's really the key indicator and what this diffusion model has been used to produce. So we can see here the time series of estimated otter abundances. And so when we're thinking about future data sets, we want to try to collect data that's going to let us minimize the variance of these estimates. We want to try to produce as precise an estimate as we can of the sea otter abundance. And so with that goal in mind, we can develop this iterative optimal design framework. So in this framework, we'll walk through the steps of it here. First thing you do is you just collect your initial data set. We'll call that Y1. Then we need to fit a statistical model to obtain samples from the posterior distribution. So here I'm using these square brackets to indicate distribution. So this would be the posterior distribution of whatever parameters we have of interest theta given our initial observed data set. So we fit that model. And then we need to generate predictions of future data. So often these will be predictions from our model, but they can be predictions from, say, expert opinion or from different sorts of scenarios you want to explore. But somehow or another, we need to generate what the future data might look like. And then we need to enumerate all of the designs that we want to consider, all the transects that we might go out and sample, and then evaluate those potential designs. And so in order to do that, we have to first refit our model. So for each design L and each likely future data set Y2L, which is what we're showing here, we need to refit the model to get our new estimates, update our estimates of theta, given our original data and what we expect to see under this particular design. Then we can compute some sort of design criterion. This will be some sort of function that captures some aspect of our understanding about the system. So often it will be the variance of some quantity of interest. And then we need to repeat that for every design. And so we can compare the designs, find the optimum, and then go out and collect the data and start the process over again. And the key here is that this step is a huge bottleneck in this procedure. This is a potentially the source of a major computational challenge here. If refitting this model is difficult, so in the sea otter context, that ecological diffusion model takes 15 hours to fit. And then if we want to look at, compare 1,000 different designs, that really quickly gets out of hand and becomes intractable. So what we need to do, and what my work has looked at, is trying to find ways to make that step of it faster. And so we can do that using this idea of recursive computing, which is a really sort of natural fit with the Bayesian idea of updating our understanding of something as new data arrives. And so in this framework, as I said, we need to obtain this new posterior distribution given our original data and our new data. And we can factor this just using probability rules into two components here. So the first component is just the data model, the likelihood of the new data under design L, and then times the second part, which we'll notice is the posterior from the first stage of the model. And so this factoring it this way actually makes clear that this posterior from the first stage is effectively a prior for the second stage. And so we don't actually need to refit this whole model. Instead, we can just update our original posterior distribution with the new data. And if you make some smart choices about how you set up your MCMC, you can do this whole thing really quickly, orders of magnitude faster than you could fit this whole new model directly. And so that really opens up this whole procedure here and allows us to effectively explore a lot more designs and apply this in a lot more cases. So if we go back to the sea otter example, so here we've collected all of the data up through 2012, we fit the model to those data, and we want to try to optimize the survey design for 2013. And so here we have to define the design criterion. So again, this lambda 2013, that is the total sea otter population estimated in 2013. And what we want to do is try to minimize the variance of that estimate. We want to try to get as precise an estimate as possible there. And so if we go through this procedure, we look at 1,000 designs of just randomly laying our transects down on the Glacier Bay. We get this histogram of our design criterion over all those random designs. And then over here, shown in blue, that is, of all those designs, this one here is the best one. And it is about, it reduces our variance, or estimated variance of the population size by about 40% using this design over just the average random design. So it does get us quite a bit more precision in our estimate of our quantity of interest. And then on the right-hand side here, it just shows what that design looks like, what those transects would be. And so this is a little bit of a toy example, but this procedure is now part of the standard operating procedure in Glacier Bay, and it's how they're actively determining their survey designs each year. All right, so just moving briefly into to talk about future work and opportunities a little bit. In particular, I'm interested in kind of focusing on these two components of this conceptual figure here, developing new mechanistic approaches, and then also trying to expand the applications of this optimal design framework. So for the sea otter system specifically, the model that we built is structured roughly like this, where we have the sea otter component, the prey component, and then we're really just using the sea otter as a forcing term to drive the prey dynamics and force the prey dynamics. But in reality, there should be a feedback loop here, and we might expect that as sea otters eat all of these really high value prey and drive them down, that potentially it might incentivize movement out of that habitat and might drive sea otters to seek new habitat that has more of these high value prey. And so we expect that there is potentially this feedback loop here. And so I'm interested in potentially developing a new sea otter diffusion model that accounts for that feedback and that brings in these different data sets simultaneously to help us better understand how the prey environment is driving sea otter movement and how those things feed into one another. I'm also interested in developing new approaches to think about capturing process misspecification and process uncertainty. So in the clam model, we just wrote down the differential equation and used that as our structure, basically as sort of a link function in our model. And so we might write down again, like we feel pretty good about the recruitment and background mortality, but maybe we don't have a great understanding of what the sea otter predator-prey interaction looks like. Previously, I used just a type 1 functional response, but it could potentially be a type 2 or 3 or a different functional form that we can't write down a priori very easily. And so there's been some work in this recently with bringing in some machine learning approaches and to sort of more flexibly just say that we're gonna say that there's another term here that's a function of both sea otter and clam abundance, but we don't know exactly what that looks like. And then we can actually estimate what that function is. And so this kind of framework lets us encode, a plausible, ecologically motivated structure in the model, but also allows for the discovery of new mechanisms. And this is really sort of more closely aligned with this idea that a differential equation and what the rules we might write down in a differential equation is really part of our prior. So it's something we want to bring into the model and draw strength from, but we also want to give enough flexibility that we can learn from the data and we can let the data pull us away from our initial hypothesis if necessary. And then on the optimal design side of things, I think there's a lot of opportunity to work on these sort of issues here in Nebraska with the state's monitoring and inventory programs. So I'm interested in particular in applying this in these multiple data set contexts. and potentially looking at the, if we have sort of this integrated population modeling setting where we have count data and, and capture recapture data, can we use this framework to look at the relative value of those different data streams? Is it going to be more useful to go out and collar more animals or to go out and do more point counts? And then similarly, in expanding it to sort of a multi-species setting, can we identify sort of keystone species for monitoring? Are there species, sort of the indicator species idea where some species might be particularly informative about the overall state of the system? And we could maybe use this approach to get at that. And then lastly, also pivoting this more towards management settings. One of the big advantages of these sort of mechanistic approaches is that we can do simulations with them. We can simulate different management scenarios and then use this iterative optimal design framework to decide where to go collect data in order to best understand what the effect of those management actions might be. So I think there could be some really interesting work to be done there that's really closely related to these like existing ideas in adaptive management. This could be a computational tool that could fit into that. And so with that, I'll just say thanks again for your time and happy to take any questions. John, Rachel. Yeah, so I guess I'll repeat for the Zoom the question of whether this framework can be used not just where and when, but to identify which parameters are going to be most important. And there's definitely, so yes, I think is the answer to the question. These frameworks can factor into that and are kind of closely related to ideas about sensitivity analysis, where you can try to pinpoint which parameters are most informative of a given quantity of interest or sort of a derived quantity like population size or presence or absence. Yeah, we could use some of these ideas to try to get at that and isolate parameters that are particularly important that way. I'll go to John last and go to Jessica next.
But the other thing that I'm thinking about is that it seems like Bayesian method kind of been on for a really long time. I remember in grad school, everyone was really, really excited about Bayesian.
Mm-hmm.
We're all and it's still sort of that was just. What do you think that we could be doing different or better? That are have such like potential beyond.
Right. Yeah. That's a great question. So the question is, again, to repeat it, is that Bayesian methods have seemed like they're kind of right on the cusp, right on the edge of breaking out for a long time now. And what can we do to kind of get over that hump and move forward with them? And I think there's a couple things there. So one is computation. It's not necessarily something that we can do in this room, but it's something that is being done to help move these methods forward. And there's been a lot of advances recently that are making a lot of these methods a lot more computationally accessible, and just sort of, just the development of computers, a standard desktop can handle most models that people would fit now. So that has been a big change recently. And sort of along with that is the rise of sort of easier to use packages, statistical software. So in R, there's sort of been the rise of Stan and a lot of packages that sort of repackage Stan into standard like GLM syntax. And so I think that is also starting to make it a lot more accessible. And then as far as what we can do, I mean, one of the things that I'm interested in doing with this position is on the teaching side. So talking with John and thinking about how we can sort of build a statistical curriculum here in SNR and with the co-op unit that can get students into using these models more easily. And so I'm hoping to develop more like a hierarchical modeling class, and so I think that's part of it that can help us move in that direction, hopefully, if it works out. But yeah.
So I understand that your research mainly concerns otter abundance and sort of comparing that with otter abundance.
Mm-hmm.
Are you also assessing other variables that might be impacting otter abundance, such as meat predation?
Yeah, that's a great question. And so the like the ecological diffusion model, yeah, so far doesn't really account for specific sources of otter mortality. It just kind of assumes the density dependent mortality through the logistic growth. But that is definitely something that my colleagues in Alaska are looking at. And even though sea otters are commonly viewed as an apex predator, more or less, there is some evidence now that they are being preyed upon by other things. So eagles can sometimes snag an otter. And then there's also been evidence recently of bear kills. And so, yeah, they're starting to kind of collect at least a little bit of anecdotal evidence that might be starting to limit sea otter abundance in some places. And then in other places, like out in the Aleutians, shark predation is known to be a major impact. But that doesn't seem to be the case in Glacier Bay so far, at least. But it could just be that the sharks haven't realized all the otters are there yet. So it could be important later. Yeah.
Seems very intuitive for you to use those priors into developing the models to time, but I'm wondering if you've identified potential pitfalls and rerunning the models over
Mm-hmm.
without the priors. Do you think there's anything that could in a situation where that would
Yeah, that's a great question. So the question again is, are there any risks or pitfalls to using this sort of recursive approach to refit these models? And there is, if sort of in the long run, if you applied this sort of over decades, if this was your scheme, you can actually get what's called degeneracy. So basically, what you're doing is you have your posterior distribution, and then you're updating that with new data. And as you do that, it can kind of shrink and shrink and shrink until it's like not actually capturing the variability left very well. And so there is kind of a need, if you're applying this over a long time scale, to kind of refit the full model, every so often, so you can kind of rebuild the necessary variation, and you don't get that sort of long-term shrinkage. And that's something that Mevin Hooten, my postdoc advisor, he's working currently on sort of approaches to avoid that problem. All right, we'll settle for John now. All right, we'll talk. All right, great. Thanks so much again.